now I'd like to give a warm welcome to our presenter today, Dr. Martha Burns, wonderful presenter for us. I've worked now with Dr. Burns for about 15, almost 16 years, 15 and a half actually to be technical here. I enjoy working with her. I love learning new things, and I learn something new every time we do one of these presentations. She is a joint appointment professor at Northwestern University and has authored 3 books and over 100 journal articles on the neuroscience of language and communication. Her expertise is in all areas related to the neuroscience of learning, such as language and reading in the brain, the bilingual brain, the language to literacy continuum, and the adolescent brain. She is also a fellow of the American Speech Language Hearing Association, ASHA, and the director of Neuroscience Education for Scientific Learning Corporation. Please help me welcome Dr. Martha Burns. Are you there, Dr. Burns?
I'm here. Can you hear me now?
Okay, great.
Sorry.
I can hear you
yeah. Thank you, everyone, for coming. That would just be a panic. Also, Carrie, for some reason,
everyone's
I
clapping.
Won't see.
Everyone is applauding, so that was the time that they needed to applaud, so you weren't over-talking anybody.
Okay, good. So if I'm not seeing your notes, so if something's going wrong, will you just pop in and tell me? But hopefully everything will go well, and welcome everyone. I'm delighted that you're here. We have a lot to cover. There's so much going on with autism all the time. It's just exploding, changing constantly. And Carrie knows we updated these slides 3 or 4 times. So I just want you to know that the very next slide that I'm going to be presenting is on the newer references since the last presentation I did. Now, some of you have never heard me present before, but these are the newer references. Many of them are summarizing a lot of the older research. So you have the reference list right there, and you can download any of those articles you want. I just want to get started with the autism prevalence. You probably know it's increasing. In the United States, per the Center for Disease Control in 1914, just last year, 1 in 68 children are now being diagnosed with autism spectrum disorder. And in some states, like the state of New Jersey, for example, it's as high as 1 in 45. And I'll explain a little bit of why that may be occurring. Some of that may have to do with diagnosing more children that wouldn't have been diagnosed years ago. But what's important to see, and you'll notice it in this slide, is that actually the identified prevalence of ASD has really increased. Just in the last 10 years, it's over doubled, really, almost tripled. So in 2000, it was 1 in 166. It's now at 1 in 68. And if you do the math, you can see that it's more than doubled. So autism spectrum disorders are highly prevalent. They are here. As I think you all know, the prevalence is much higher in boys. Actually, we have 1 in 42 boys are diagnosed with autism spectrum disorder. That's 5 times more common than girls, 1 in 189 in girls. The prevalence of diagnosis is higher among whites than it is among blacks. It's a third higher, and it's 50% higher in whites than Hispanics. And we'll talk a little bit about that. Some of that, again, may be that the fact that children coming, let's say, from Mexico might have been diagnosed with autism spectrum disorders had they been here when they were young, but if they emigrate later on, they may not. And parents who are more likely to go to doctors or psychologists to get the diagnosis might have a higher percentage. We're not quite sure why that would be. But what is going on? What do we know? Why is the prevalence increasing, and what do we know? The 2 that you see up here first are that autism spectrum disorders are one of several neurodevelopmental disorders. So there are other neurodevelopmental disorders. Lots of them are increasing as well, not at the same rate as autism spectrum disorders, but other genetic disorders are. We also know, and we've known for a long time, that autism spectrum disorders are very heterogeneous disorders, that there's a lot of diversity in severity and symptoms. And what I'm going to do as we go through this is show you that that has really increased even in terms of our understanding of these disorders. There is no one autism spectrum disorder, so there's not going to be one cause, there's not going to be one set of symptoms, and we need to keep that in mind. Now, some of the newer findings that are out that I'm going to share with you, and we'll go through each of these separately with separate slides, is there are patches of cortex that are disrupted. We have several studies now showing that. We have research that was done by Martha Herbert. We have other research that was done by Stoner et al., and they have found little regions of the cortex where neurons either didn't migrate to the right place during the stage of migration, which is the second trimester of pregnancy, or the neurons for some reason are just not developing, and it could be synaptic problems, which I'll explain more in a minute. Secondly, there are problems with brain connectivity. And that's huge, and that's brand new research in a certain way. We knew for a while, we've known probably for 7 or 8 years, maybe even 10 years, that that when we look at certain kinds of images, diffusion tensor images are one good example where you can see white matter tracks, that the white matter tracks, if you look at children on the autism spectrum, are different. Some of the longer white matter tracks are not maturing well, and some of the shorter, more local fiber tracks are over mature, if you will. So there seem to be problems with connectivity. We've known it for a while. We're starting to get a better sense of that right now. We also know that there's an imbalance in excitation and inhibition, and I want to explain a little bit what that means. What that means is that, and we're going to talk about this more in a minute, that when when two neurons connect, as you know, they have a synapse, and that's how neurons communicate with each other, and we'll kind of review that again in a few minutes. But they're not always excitatory. So sometimes synapses are inhibitory, and sometimes they're excitatory. So I'll give you an example right now. Right now you're trying to listen to me talk. And there's a lot of distractions when you're listening to somebody over the Internet. You may not have the best reception. The slides may not be coming up exactly the way you would like them to. And so those things can be distracting. And you might even have something going on in wherever it is that you're listening to me. Maybe you have a child in the background, or maybe you're in a school, in a classroom, and there's something going on outside. And you have to ignore those. So all of that information that's coming into your brain that you want to ignore has to be inhibited. And then what you want to pay attention to has to be excited. That's what you want. Those are the stimuli that you want to get through. And if that balance gets thrown off, you're highly distractible. The world maybe seem very noisy to you because all the stimuli that are coming in excitatory, and you're not able to tune anything out. So we've known for a long time, actually, Michael Mersnick and others started to identify this 15 years ago or so, that there's this imbalance in excitation and inhibition. And I'm going to explain now what they think is going on. There are two factors that may lead to this, to the connectivity disorders and to the problem with excitation and inhibition. The first, it looks like the brain doesn't prune itself very efficiently or as efficiently when a child has autism spectrum disorders as when a child is neurotypical. And we'll talk about pruning more later, but just think of pruning the way you would think of pruning a rose bush or any other plant that you have that you want to bloom. You prune it back so that the bush has energy to make the blooms. So if you have a rose bush, you prune it way back in the spring, and then get all the long tendrils off, and then that gives the rose bush the energy to produce the roses. Well, the same thing happens in the brain. You want to sculpt the brain and prune it so it's good at some things and not so good at other things that you don't need. And if there's either over pruning or under pruning, just as you can interfere with a rose bush blooming, you can interfere with the brain blooming and developing in a typical way. And then finally, there are multisensory integration problems. The brain doesn't integrate itself as well as it does with children who are neurotypical, meaning that if you're trying to listen to me and you're trying to watch the slides at the same time that you're listening to me, you need to integrate those two bits of information. And if you can't do that, then that becomes frustrating and anxiety producing. And OTs for a long time have known that there were multisensory integration problems. It's why they do something called sensory integration therapy. And they've noticed it in the motor and tactile world, so the children are tactily hypersensitive. We're now starting to see that it cuts across all modalities. So it also affects language and other aspects of cognition. The big story right now is that autism spectrum disorders is polygenetic. And what that means is that there are complex inherited and mutant changes in the genes. So the genes mutate. They have changes associated with them, and those often have names like copy number variations. And I'm going to explain what those are. We all have genes that are mutating all the time. Our genes replicate themselves constantly. Every time a cell divides, it's going to replicate the DNA that's within that cell. All of our cells have all of our DNA, and errors can be made when that DNA is replicated. And when those errors are made, that's a mutation. Now, we all have mutations. They happen all the time. Mutations are associated with a lot of things we don't like cancer, for example, but they're also associated with things that are adaptive. So the fact that many of us have been able to consume cow's milk and not be lactose intolerant has occurred because we had mutations that allowed us to digest cow's milk. So mutations occur in all of us, but when the mutations are a certain kind, in certain kinds of chromosomes and in certain kinds of genes, that predisposes children to autism spectrum disorders. And then there are some inherited genes that we call autism risk genes. So with that, let's talk about this whole idea of the synapse, and let's talk about what it is that the research is now showing about this problem at the level of synapse excitability. So in this particular slide, what you see is that you see 3 neurons, and those are those round circles that you see at the bottom. You see what looks like a big long tube, a big long hose, and that's a blood vessel. You see little red cells, and those are called astrocytes, and those deliver nutrients from the blood vessel to the cell body of the neuron. And then you see these neurons. And neurons have long fiber tracts that send information. Those are called axons, and little fingers that stick out to pick up information. Now, where the 2 neurons meet is called a synapse, and that's what you see in the upper right-hand corner. And the way a synapse occurs is that the axon has a little tip at the end of it, and inside of that are little bubbles, and you can see those in that inset. It says synaptic vesicles, and they're little bubbles, and I want you to think of those like you would think of the pulp of an orange. They're going to be filled with neuromodulators or neurotransmitters. And then you see on the receiving end at the bottom the little receptors sticking up on the tip of a dendrite, and those are going to suck up the neuromodulators that are released, or the neurotransmitters that are released by the axon. So if you squeeze an orange, you squeeze the pulp breaks, the orange juice comes out, and then you can drink it. Well, that's exactly what happens in the brain. Now, and just as one other reminder, sometimes synapses are excitatory, so it allows you to watch what's going on or hear what's going on, and sometimes they're inhibitory, so it allows you to ignore things you don't want to pay attention to, either stimuli coming in from your body, maybe your stomach's growling, or stimuli coming in from the world around you. If there's an imbalance in these synapses, if there's too much excitatory activity, too much excitatory transmitters, or too many inhibitory transmitters, then what you do is you create an imbalance, and the child might tune out way too much and not be able to pay attention a lot, or the child might be hyper-attentive. That is, they might be paying attention to everything going on without them and not being able to fill it out. And that's where the new research is going right now. Now, one other thing that I just want to remind you of is this slide. And this is just one network. It's called the Object Naming Network. And it's to remind you that regions of the brain work together, just like your cell phone network. So this is the Object Naming Network. It has neurons in the temporal lobe. That's that bottom hump at the bottom of the brain. It has neurons in the frontal lobe. That's the front third of the brain. And it has little neurons up at the parietal lobe. That's kind of the top section of the brain. And by the way, the very back of the brain is occipital lobe for vision. But for naming, for language, we have these regions, and they're all firing together. And we often say neurons that fire together wire together in networks. Now, you have thousands, probably millions of networks in your brain. You have networks that you use to listen to language. You have networks that you use when you're speaking. You have networks that you use when you're driving. You have networks that you use when you're watching TV. You have networks that you use when you're writing a paper. You have networks that you use when you're doing a video game. Everything you do requires different networks. And what the researchers are starting to see is in autism spectrum disorder, networks are competing with the networks that we need to do things like talk and communicate and pay attention to social cues. So we have social networks that help us to watch someone's facial expression and tune into their vocal intonation. We have language networks that help us to interpret what they're saying. We have reading networks. And then we have all these other competing networks. And in autism spectrum disorder, it looks like some of those networks that the rest of us don't use a lot are being used by these children more often. They're competing. Now, what happens is the brain is a use it or lose it organ. So when networks start competing with, let's say, a language network, and the child uses them all the time, then that will surpass the power of the network, let's say, for language or social skills. So a child might get really, really, really good at one splinter skill or two splinter skills, which is a different network. Let's say if you think of a specific talent that some of my children on the spectrum have. I have one child who can sit down and play any piece on the piano he hears. He can play Mozart without even having to look at the notes or anything. He can just hear it and walk over to a piano and play it. That is a network that's very strong and very powerful. But what that's done is it's competed with his language network, and so he doesn't talk very much. There's another little child who's really good at drawing. He can fly over London. He's been on YouTube. You might have seen him. He can fly over London. He can actually, or over Athens, Greece, I think. He can actually fly over it once and then spend 3 days just drawing every building, every window, every car that he saw. But what that means is that network is overriding the other ones. And so language doesn't get built, and that's kind of where the research is going now. So let's look at each of the studies that has come out that's in your bibliography that's new since my last talk. The first one is the research by Stoner et al. That showed these cortical irregularities. This was published in 2014. And these are neuropathologies in the cortex, which is the outer layer of the brain, the part of the brain that you see here that has all those grooves. And the cortex is where all the cell bodies of the brain are, all the neuron cell bodies. And what Stoner found was in the dorsolateral prefrontal cortex, that area where you see that first square on the upper left-hand corner, and the top of the temporal cortex. It's just that second square is sitting on top of the temporal cortex there, just is over it. That's where he found some irregularities. Now, other people like Martha Herbert, who's at Harvard, has seen irregularities on other kinds of tests. He actually took post-mortem samples of brains of autistic individuals after they had died as adults and just looked at their cortex, But what Martha Herbert actually did is specific kinds of scanning, and what she found was little regions all over the brain. So he found these in two key regions for communication and language and social skills. Martha Herbert says, and her colleagues say, they've found them kind of distributed all over the brain. So it looks like the cortex has some regions that don't develop in a typical way. And the cortex is where all the work goes on in the neurons, and so that could explain why some networks might be stronger than others. You might have networks where you just don't have as many neurons activated. Now, the history of the connectivity problems in autism spectrum disorders, there are several good studies of that. This is just one slide that I reviewed from a paper by Wolf in 2012 on the last talk I did, just showing this is called diffusion tensor imaging, where you actually look at axonal fiber tracts. And what I want you to notice here is, and he has actually 9 of these fiber tracts, big longitudinal fiber tracts, long fiber tracts that went from one side of the brain to the other or from the back of the brain to the front. And the red lines are the children with autism spectrum disorder, and the blue lines are the children who are typically developing. And what he found was that there's connectivity early in development at 3 months. You can see that 3 at the bottom of this slide. But by the time you get to 27 months, when the child is 2 years of age or getting on to 2.5, you see that the connectivity has leveled off. It doesn't continue to grow, and it should grow with experience. So that was the early connectivity problem that we've been seeing for a while. In addition to these regions of the cortex that seem not to be developing, we've known since Michael Merznick's first paper in 2003 on excitation and inhibition that the brain also just isn't firing at the right rate. It's either overfiring. Some of these synaptic connections have too many excitatory neurotransmitters. There's too much excitation going on in regions that you don't need that much, and there's not enough in others, or vice versa. What I've shown you is a paper from Bergeron that just came out this last week, and it's in that bibliography of references. And you can see how complex synaptic functions are associated with autism spectrum disorders. This is that kind of inset you saw in the other picture of the synapse. And what he's showing, what Bergeron is showing, is all the different things that can go wrong with the synaptic connection in autism spectrum disorder. And obviously I'm not going to try to go through with you. That is biology that none of you really need, but it's important to see how complex this whole issue is at the level of the synapse. Now, with respect to pruning, just a reminder that the brain sculpts itself. So the human brain, as we're maturing, is use dependent. Things that we do, and we do a lot, get better and better and better. We get better and better at those. And things that we don't do a lot, we don't get better at. And that sculpting process is very important for fine-tuning your brain to be good at what you need it to do. So, for example, in our society, we don't track animals, and so most of us have not developed the capacity to look for details when you're going through the forest that might suggest where animals have been. And those networks just wouldn't have developed in us. They would if you were a Native American and you were still tracking animals. They would if you were living in a society in Brazil or in Africa where you were still hunting animals. But for the rest of us, those aren't going to develop. We're going to develop networks for reading and for language. And that pruning is important, and we have a lot of research that shows there's very specific kind of gene. It's called the mTOR-dependent system, and that is one of the genetic alterations in autism spectrum that can lead to these pruning problems. And the pruning problems seem to also be related to the synapses. So if you have a part of the brain that's more excitatory, being more activated, then that part of the brain won't prune itself. If you have a part of the brain that the child's not using, let's say they tune out every time you talk and they don't pay much attention to language, and they have trouble tuning into language, then that's going to be way over pruned. And so those pruning deficits are an issue. And then finally, we know that there are these multisensory integration problems. We've known, as I said before, these have been identified by OTs, occupational therapists, for a long time, that children are tactily hypersensitive, that they have trouble integrating what they see with what they feel. And there's just some research that's in that reference list by Fox that came out in 2013 for speech-language pathologists among you, showing that in speech we see that children cannot integrate what they see, the face and the lip reading and the kind of watching facial expression, with what someone's saying. And so that could lead to problems with pragmatics. That leads to problems picking up social cues, understanding things like sarcasm, and that then is one of the major problems. Now, just briefly, this whole issue of genetics, because all of these issues seem to be related to genetics. The genetic risk of autism increases with family history. So if there is a family member who has a prevalence of autism spectrum disorders, and a cousin or an aunt or an uncle, we're much more likely to see it in the family. So we know that we have something called autism spectrum disorder risk genes that exist in some of us. And the second thing is there are these rare genetic mutations that are seen in individuals on the spectrum. And I'll talk a little bit more about that, but they are in very specific places, and they predominate when you have a child on the autism spectrum as opposed to when you have a child who is not on the autism spectrum. And then this is where it gets really complicated. But some of the genetic mutations seem to be specifically related to neural connectivity. So some of them seem to be causing these problems with synapses and the plasticity, or the changes in the brain, the pruning in the brain that result from synaptic connections. Now, these are complicated slides. This is one on the genetic risk. I don't want to spend too much time with it. You can get it from the article. You can also get it when you get the slides. But essentially this is Borgeron, and essentially what is being stated here is just on the right-hand side, you see that the more you have a genetic relationship with someone, or the closer your genetic relationship, let's say you're an identical twin and one has autism spectrum, the more likely you're going to see autism spectrum disorder in the other twin. The further you go away in the family, the less it is your immediate family, let's say cousins, uncles, aunts, then the risk goes down. And the slide, or the part of the slide on the left, is just showing that when you look at autism spectrum disorder kinds of tests, that what you see is that there's a normal bell curve of the general population. If you look at the bell curve of relatives of people with autism spectrum disorder, you just see it shift a little bit more toward the symptom side, and then, of course, it would be higher among children who've been diagnosed with it. Now, just understanding these genetic mutations very quickly, I don't want you to feel like this is a course in genetics, but the causes are caused by this replication, that genes are constantly replicating themselves. So every time any cell in your body divides, you're going to have a gene that replicates itself, and those gene copies are called alleles. So if you're ever reading about this and you read about these alleles, that's what they're talking about. The errors occur when any number of things can go wrong. There are lots of potential sources of errors in replication, and the cancer, as I said earlier, is one example where you have, as you age, when the genes replicate themselves, they're more likely to make mistakes over time. And if they make a mistake and your body doesn't pick it up, that can be the beginning of cancer. But there are de novo mutations that occur, and de novo just means anew. So they occur for the first time, and they occur in either the mother's egg or in the father's sperm. And those are just the maternal or paternal cells. And this little mutation occurs, and if it's in specific kinds of cells during brain development, then you can start seeing the mutations that are associated with autism spectrum disorders. One kind that's very common in autism spectrum disorders is called copy number variations. And that's just, you can see the little gene on the left duplicated itself, and when it did one part of the gene replicated itself twice. So you have a copy number variation. You now have two copies of that section of the DNA. And when we see these copy number variations, either in the inherited genome or when we see it caused by de novo mutations in the father or mother, that's what seems to contribute to the risk of autism spectrum disorders. Now, I know some of you are parents, and I just want to, for you and others who are interested, if you're interested in all the genetics and all the research going on and what's causing some of these mutations, there's a wonderful website. It's the sfari.org. So it's genesfari.org website. I put it up here for you. When you get the slides, you can go to it. And in this particular part of the website, you can click on any of the human chromosomes. We all have 22 chromosomes plus the sex-related X chromosome, where a lot of autism spectrum de novo mutations occur. The two sex-linked are X and Y. But you can click on the chromosome anywhere, and wherever you see those little red X's, those are areas where autism spectrum de novo mutations have been found and occurring. And again, this next slide is fairly complicated, and I don't want to spend too much time with it, simply just to let you know that they now can pretty much predict, and all of us can have our genome mapped if we want, to kind of predict what the chances would be of having rare mutations and the relationship between rare mutations and your own genetic inherited background. But what you can see in the little men over on the right is that you can inherit autism spectrum disorders directly just through the fact that you have the mutation in an individual. We also have what are called genetic buffers, and those are genes that actually counteract the effect of an autism risk gene. And some of us might have buffers, and that might help us not to have a child on the spectrum. So you might have one person who has a risk gene in their family, but another person who has a set of buffer genes. And then you can have all of these mutations that occur that we don't even know why, and that can be the issue that leads to autism spectrum. But the reason that I bring this all up, and this is what's so complicated, is that the neuronal genes themselves are the largest. So the brain cell genes, the genes that we have in our brain cells and that regulate brain cell development, are very large. Secondly, neuronal activity that regulates the ASD risk genes and the proteins they occurs in the brain. So here you've got the part of your body that is controlling the genes themselves, and whether they're expressed and how they're expressed are your neurons. So you've got neurons that are most vulnerable to mutations, are the ones in the brain, and they're the ones regulating the way the body responds. And the proteins, those neurotransmitters you saw that determine whether it's excitation or inhibition going on, those proteins are what modulate the activity of the synapse. So if you're going to have a genetic mutation, the chances are much higher that it's going to affect your brain than any other organ in your body. And once it gets dysregulated, that dysregulation itself seems to cause some of the problems that occur. And the reason this is also important then is the key to intervention, and that's where I'm going to stop all of this and just talk about what we can do about it, is getting the brain regulated. And the way you get the brain regulated is by actually counteracting the effect of this either synaptic upscaling that you see where the brain is overplastic, or the genetic downscaling where the brain is underplastic. And even though that sounds very biological and crazy in a way and very complicated, really what you're doing is saying, okay, let's get the brain to do the things we want it to do. Let's get it to do the behaviors that are adaptive, that we know are regulated. So if I have a child who is on the autism spectrum and they love more than anything else to get onto an iPad and play with an iPad all day, the last thing I want to do is let them get on the iPad, if that makes sense. Because what I'm going to be doing is I'm going to be creating more excitation in those very fiber tracts and networks that are competing with language and competing with social skills. So the key to getting the brain under regulation is getting the child to not do what comes naturally, not do what they love, which is the hardest thing in the world if you're a parent or even a therapist or a teacher, but to get them to do what balances out what they love, to get them to do what they love is competing with. Now keep in mind, ASDs, as we said, are very heterogeneous, and so we don't want to do the same thing with every child. One of the things we have to do with these children is we've got to figure out what their strengths are and what are the networks that are competing with language, with communication, with social skills. And so we're going to have different problems with different children, and it's going to have to do with the synaptic plasticity and the way the brain is pruning itself. So that what we really need to do is get the regions of the temporal lobe and the frontal lobe to be more activated and underplay or understimulate regions of the brain that are competing. So what can be done, again, just to summarize this and to take it where we are, and then we'll take it to the evidence-based treatments. Well, number one, behavioral treatments like ABA, the applied behavioral analysis, are excellent if they're individualized to the child's specific problems. So if I have a child who isn't responding to speech, the first thing I want to do is start doing a lot of repetitive practice and reinforcing constantly to stabilize those networks. And you stabilize networks through a lot of repetition and then reinforcement, and that's what ABA is all about. Applied behavioral analysis. Exercises then have to be repetitive and compete with non-adaptive networks. So I don't want to feed in to what the child is doing all the time and loves. And that's hard. So if my child really loves dinosaurs and all they want to do is read about dinosaurs and think about dinosaurs and learn about dinosaurs, or they love Thomas the Tank Engine, I got to break them away from that. And I've got to introduce other things for them to be interested in. Or what I'm going to do is build up the dinosaur skills to the point that they're crowding out the rest of the knowledge that child needs to do well in school or to do well with friends. And then finally, we have neuroscience design programs which were specifically designed to create homeostasis, specifically designed to stimulate all of the important networks and understimulate networks that don't contribute at all. So you want to stabilize the adaptive networks, the networks for attention, for social skills, for language, and what you want to do is improve what we call homeostasis or balance in the brain, and those are very important. So we have research that was done a few years ago looking at neuroscience-based intervention, the Fast Forward program by G. Poglich and Marcy Melzer, where they talked to 34 professionals and got data from 34 professionals across the U.S. who had run children on the autism spectrum through Fast Forward for at least 20 days. They had to have been diagnosed with autism spectrum disorder by a medical professional. So they couldn't have had another diagnosis like PDD-NOS that was diagnosed by someone who wasn't a medical professional. And they looked at age equivalent scores, and they looked at a functional skills checklist. And number one, they found with their response rate that 81% of the children who had been on the Fist Forward program actually showed an increase in their intentional skills. Attention for testing, for computer activities, for therapy activities, for listening to stories, for group activities. And I want to show you just a demo. I'm hoping you'll be able to hear this all right. I've got everything up the way it's supposed to be. I'm going to put myself on speakerphone, and I want you to just see a task that a child has to pay attention. In this case, they're paying attention to speech sounds. They have to hear speech sounds in words like bat, pat, mat when they click on these speakers, and then they have to find the match. So it's actually working on attention and memory. Put on speakerphone and play this, and hopefully you'll be able to hear it okay.
Tub, tub, cut, dug, dug, bud, buck, butt, cut, buck, buck, bud, bud, butt, butt, cut, cut.
So as you can see there, that's a kind of exercise you wouldn't see anywhere else. It's just designed by neuroscientists to improve working memory skills, auditory attention skills with thousands of repetitions. The child does that for 20 minutes. They have to maintain their attention. They have controlled reinforcers. Now we want to build in language skills. And what the research found was that 83% of the 100 children that were looked at showed improvement in language skills. Remember, this is over just 20 days of treatment in some cases. They showed language response time, improvement in following directions, understanding humor, overall comprehension, imitation, vocabulary, answering questions. And I just want to show you an example of how that's worked on with a neuroscience-based intervention. And what you're going to see here is when we get started, the child is going to have to figure out which picture goes with which kind of grammatical sentence in this case.
The wolf is running with the basket. Little Red is walking with the wolf. The wolf that is not chasing the bird is drooling.
Now, what we want is more complex language for older children. We want to also pair that with reading to build up these language skills. Remember, you're just trying to exercise these networks. Again, these children are doing these for 15, 20 minutes at a time, getting lots of repetition. Here's an example of where the child doesn't listen to the language but has to integrate the visual and the auditory for reading as well. And I'll read it when you see a word you don't know. Look it up in the dictionary. You can find words starting with A at the beginning and words starting with Z at the end, and you'll find a word you can't read its definition. This tells you what the word means. When do you use a dictionary? The child has 4 choices when you don't know the words. How are words in the dictionary ordered? Letter of the alphabet, I'm sure you know that. So here what you have is both comprehension and you have language being built all at the same time. And then this is just an example of a higher level. That was about a 2nd to 3rd grade level. This is a 3rd to 4th grade level reading comprehension exercise that you can see. And in this case, what you're looking at is a graph, and the child has to look at the graph, and then they have to figure out which museum opened in 1969. Where is Old Sturbridge Village located? So you can see the language gets more and more complex as the children go through these exercises. How many museums in the table are not in California? In 1940, how many of the museums were open? So the integration, the multisensory integration that's important here, integrating visual, integrating language, and being able to read charts, that's all important. And then finally, when you build homeostasis in the brain. And when you create networks that are positive for vision, for language, for attention, for memory, you see increases in social skills. So 76% after just 20 sessions in some cases showed improvements in eye contact, addressing people by name, being able to maintain a topic during a conversation, appropriate attention-getting approaches, using greetings, refusing in appropriate ways, or protesting, ability to negotiate and initiate conversation. And then we have research that was published by Russo, Hornickel, Nickel, Zecker, and Krauss out of Northwestern University just a few years ago, where they actually looked at auditory processing skills in these children. And what they found was even at a very low level of processing, the fast-forward exercises fine-tuned the ability of the synapses to respond to speech and to be able to perceive speech in noise. And then we see in general, not children on the spectrum, but children who are struggling readers. We've seen research that was done at Stanford University, replicated at Harvard a few years later, actually showing that you activate those regions of the human cortex that are underactivated in children who are struggling with specific skills when you use these neuroscience design programs. The programs are Fast Forward and Reading Assistant, and this is the information that you can go to get a demo or to email questions. And now I'll turn it back to Carrie for questions that she can ask me.
Great. Thank you, Dr. Burns. Just to make an announcement to everybody, some of you folks noticed that there was a little bit of a discrepancy between which slide Dr. Burns was on and what she was talking about. Not to worry, for the recording, that will not be an issue. So if you do want to view this again, you won't have that issue to view the recording, just so you know. And the slide, certificate of attendance, and a link to the recording will be emailed to you within the next few days. After we're done today, there will be a post-webinar survey page that will come up, and it would be great to hear feedback from all of you. Plus, you'll see a link to the certificate and to the presentation slides on that post-survey page. So please, after the meeting ends, please look at that page. That'll pop right up. So now we're going to go to questions. And this was an interesting question that we received just a few minutes ago. And let me find it one more time. It was about how does one I think it was, and I have to find it, but basically it was about how does one understimulate the brain? You had mentioned understimulation of the brain.
Sure. So, understimulation of the brain occurs when you're asleep, your brain is resting. But when you, it's a balance. The issue is balance. Mm-hmm. So if a child is tuning out, let's say, and I've worked with hundreds of children on the spectrum in my fortunate, but many, many years of practice. I've been in clinical practice for over 40 years. And so I've worked with many children on the spectrum. And one of the things that you often see, especially when they're young, is they don't pay attention to speech. They tune you out. They just aren't. You say Billy, you say their name, it's as though you're not even talking. They might not even pay attention to my body. Sometimes the child will actually literally, if I'm sitting on the floor with them, will walk on me. Now that's understimulation. The brain is not being activated for speech, and it's not being activated for visual stimuli in the environment or tactile stimuli. So that child is under-responsive. Then you can have hyperactivation where the child's over-responsive. So maybe they're over-responsive to loud noises or they're over-responsive to any visual changes that go on. So what we're looking for is a balance. But when I said under-excitation, what that means is those parts of the brain, those networks are not firing enough. You're not getting enough neuromodulators in those networks. The synapses are under-activated, and the child is tuning that information out. That's not getting into the brain. I hope that explains it.
Okay. Next question that came in is, what about motivational assessments? A lot of the time what motivates the students will be the thing that we should try and get away from so the brain can develop in different areas. For example, we do a motivational assessment where dinosaurs are most motivating. Do we still use it as a reward?
Yes. So that's a wonderful question. Whoever asked that gets an A+, because that's what you want to do. You want to do what the child loves as a reinforcement for finishing or doing what they don't love. So if a child loves dinosaurs, but he really doesn't like to have to sit and watch me and follow directions, then what I'll do is say, okay, you will follow directions for 10 minutes, and then when you're done, you get to play with that brand new T-Rex that I just bought yesterday, and he'll get to play with that. So you use what they love as a reward, and that's fine because they love it, and that's okay. But you don't let it monopolize their day or even an hour or even a half hour, because that network will become overactive. That will compete with the other networks you're trying to build.
Wonderful. Then the next question is, I'm always looking for better research-based activities that can be used with younger preschool children, preschool-age children in early intervention programs. Any new suggestions for way younger children?
The newest research on the preschoolers is on prevention, actually, and there's a lot going on at UC Davis, but essentially what they're recommending for the really young children is talk to them, play with them, but try to get them to avoid computers in terms of like just playing and working by themselves and doing things by themselves, and try to get them to avoid their own kind of repetitive internal things and interact with the parent and interact with adults and other children. So anything that gets the children moving in that direction, and I would go to the UC Davis research because they've really done a lot with the younger children, starting at about 18 months up to about 3.5, 4 years.
So is it, and this is more my question also as well, so we've heard the research that it's important to interact with young children anyway instead of putting them in front of computer screens or TVs. Is it even more critical with students or children with autism that they have more real interactions? And
yes, and the hallmark is to get them to do what they really don't like to do as much. So if they love playing with Thomas the Tank Engine, buy other toys, other trucks, and sit down and play with them on the floor and interact with them. And then, of course, the other thing is ABA. Applied behavioral analysis is excellent for the younger children to get them to be able to sit, to get them to be able to respond to an adult. But I wouldn't say the 18-month-old. I'd say more about the 2-year-old, 2.5-year-old for ABA.
Okay, wonderful. Let's see here. Next question. This was good. And this is on the other end of the spectrum. Is any research being done in the area of early onset dementia and the use of fast forward programs that can help maintain language areas of the brain that are falling out of homeostasis?
Wow, that is also an excellent question, because it looks like many of the same genetic changes, and I won't say the same genetic changes because it just happens to be the same genes, because so many of the genes that have de novo mutations and that mutate and have copy number variations, so many of those genes are neurology or brain genes. But the genes are changing in Alzheimer's disease too. And so Michael Mersnick and the programs that used to be called Posit Science, that's now called Brain HQ, was actually designed to see if that kind of stimulation can reverse the changes that are occurring in Alzheimer's disease. It's very hard to test that because Alzheimer's disease does progress. And so it's hard to see, are you slowing it down, especially once you've identified it. So again, the research is going to prevention. If you have Alzheimer's genes inherited, can we prevent the onset with cognitive stimulation? And there's tremendous research going on in that area.
Okay, next question, and this is actually a really, really good question. We haven't really delved too much into this area on our webinars. How does PTSD affect the connections in children on the autism spectrum in general?
Oh, my goodness, we have a smart audience here. Wow,
I know,
you guys
really
are asking all the right questions. Yes.
Really good questions.
Yeah, so kids on the autism spectrum often have anxiety disorders, and being on the autism spectrum is stressful for the child because the world is scary and disorganized and noisy, and it's stressful for the parents. And there's a great deal of research now on what stress does to the development of the brain. So I didn't even get into the whole mood kind of side of autism spectrum disorders and anxiety disorders. That Bourgeron article does get into that quite a bit because a lot of those de novo mutations are affecting things like anxiety and mood, and they do overlap very often. And there's quite a bit of research on how to down-regulate that anxiety because that will also take over, and that will short out development of the cortex. So getting the children regulated, and that's really where OT is very helpful. Occupational therapy is very helpful in creating, and ABA again, in creating this kind of behavioral world of being more relaxed and being more adaptive from a mood standpoint. And then also there's a lot of research right now in medications. There's nothing that I know of right now that's out there, but a lot of research being done with animal models to see what can reduce that anxiety, because it's hard to know sometimes with a child on the spectrum how much of their tuning out or rejecting us is because of anxiety and how much of it is because of the brain not responding to those stimuli. So quite a bit of research in that area. It's a whole area in and of itself, actually. Excellent, excellent question.
Okay, I've got time for one more question, and just a reminder, as soon as we're done and I close the meeting down, you will get a post survey page and or post webinar page that will give you a survey. How did you like the webinar, et cetera. And also you'll see links to our certificate of attendance and the presentation slides. So one more question, and this is a really good question. Has there been any research indicating causes of DNA mutations? Stress and environmental toxins come to mind. Can you provide a research direction, and what prenatal research has been done?
That also is a $64,000 question. One of the references that's in that reference list is Martha Herbert, and also the Bourgeron article that just came out this week that's in Nature Reviews Neuroscience. Both of those discuss, to some extent, causation. Martha Herbert is much more interested in causation. You have UC Davis has a whole program called Marbles. There are other programs around the country looking at causation. And it looks like it's multiple issues, but certainly pesticides in the environment seem to be one issue. Stress seems to be maternal stress, because when a mother is under stress when she's pregnant, it can cause mutations. Exposure to byproducts of oil refining have some neurotoxic effects. So right now, I will say it has nothing to do with diet so far. There's been no evidence that diet of any kind is contributory, but there are neurotoxins in our air that never were there before. Maternal stress can be an issue, and there are other byproducts of just creating synthetic materials and refining oil that may account for some of the increased incidence in states like New Jersey or Texas. And there's quite a bit of research on that. So, yeah.
Wonderful. Yeah, there is. We are out of time today. We are at an hour here, and please send me any additional questions at webinars@scilearn.com. Again, I'm going to close the meeting down, and a page will come up with a post-webinar survey along with a link to a certificate of attendance and a link to the presentation slides. So thank you for joining us, and I look forward to seeing you all online again soon. Bye-bye.
Thank you, Carrie. Bye-bye.